Okay, let's start the cardiovascular system. Cardio means heart, vascular means blood vessels, and then we have blood in our blood vessels. The heart is a pump that moves the blood around the body. This is quite a big system to cover. So we break it up and we talk only about blood, then only about the heart. And then when we get to the blood vessels, we add in the blood vessels and blood pressure and all that good stuff. Okay, so let's talk about blood. So we have vocabulary over here on the right-hand side, a whole blood sample. If someone says a whole blood sample, it is a sample of blood that they have taken. You take a sample of blood, that this in this test tube is a whole blood sample. Now, if we put it in a centrifuge and we spin it around and spin it around and spin it around, then the heaviest things will go down to the bottom. And those heaviest things happen to be cells called erythrocytes. They're called red blood cells, but in this class, we'll call them erythrocytes. And they are the oxygen carrying cells in your blood. They carry the respiratory gases or oxygen mainly. So when we spin a whole blood sample and the red blood cells go to the bottom or the erythrocytes go to the bottom, this bottom portion here is just made of erythrocytes. I'll tell you what formed elements are here in a second. And this is the hematocrit. It doesn't include anything else, just red blood cells. So the hematocrit is the percentage of a whole blood sample of erythrocytes. Probably be the last time I write erythrocytes. It's much easier to write red blood cells. So in a whole blood sample, we spin it. The heaviest things go to the bottom. Those are going to be the erythrocytes. And then sitting right on top of the erythrocytes will be a small thing called a buffy coat that only represents really 1 to 2% of the volume. That should say 1 to 2% of the volume of whole blood altogether. What is the buffy coat? I should have had that listed in here. The buffy coat will be the white blood cells and the platelets. So let's get these crossed off our list. White blood cells are called leukocytes. For red blood cells, we call them erythrocytes because erythro means red. Leuco means white, cyte means cell. So white blood cells, these guys are for immunity. Here, the erythrocytes are going to carry respiratory gases, especially oxygen. Leukocytes will be for our immunity to help fight off infection. And then our platelets are the thrombocytes. Most people just call them platelets, but it's good to know the word thrombo because thrombo means platelet, and it'll come up quite a bit when you're doing the reading, like the thrombin, prothrombin, etc. You'll hear this term. So thrombocyte means platelet, and this is for clotting. Later on in the lecture, we tell you exactly how platelets are involved with clotting. So this is our whole blood sample, everything in a blood sample. The hematocrit is only the red blood cells. The buffy coat, which I should have put in here, that would consist of your white blood cells and platelets. And now what is this termed formed elements? Formed elements are all the cells in blood. So this is a formed element. Erythrocytes, leukocytes are formed elements, and platelets are formed elements. Here it looks like how they labeled this, that the erythrocytes are only the formed elements, but no. Platelets, white blood cells, and red blood cells are your formed elements. And this gives you a percentage a number per cubic millimeter. That should be a 3. And so platelets, notice we have 250 to 400,000. Then when it comes to white blood cells, look how few white blood cells there are compared to platelets, 5 to 10,000. And then when we get to the red blood cells, we're talking about millions per cubic millimeter. So keep those numbers in mind. You don't have to memorize these numbers, or not for me, you don't, but just their relative percentage. All right, we've got some other vocabulary words here, but we'll get to them here in a second. Notice that plasma makes up 55% of your whole blood sample. Plasma is a liquid portion of your blood. And then if we break down what plasma is made up of, well, mostly plasma is water. But then we have a lot of proteins. That's the next highest percentage by weight. And then we have other solutes like ions. Ions are electrolytes. They're anything that carry a charge in solution. Sodium, potassium, magnesium, calcium. Those are your ions. Nutrients from the digestive system. We eat, the food goes into our digestive system, and then the digestive system breaks down the food, and we get the nutrients and put those into the blood, and they're carried around in what part of the blood? The plasma. Waste products. An example of a waste product that would be carried in blood would be nitrogenous waste. If we take in proteins and eat proteins, then we have to get rid of the nitrogenous waste. We're going to do that with our kidneys, but that would be an example of a waste product, nitrogenous waste. Or another example would be like, this would be counted as a gas and as a waste product, but carbon dioxide is a waste product that we make by all of our cells, and it's carried in our plasma. And regulatory substances is a very vague term, but think of maybe hormones, something along those lines, I guess. Proteins. What are the types of proteins that we have in our blood? We have fibrinogen. Now this is going to be, it's the inactive form of fibrin. Fibrin is going to form a mesh net that's going to be the basis of our blood clots. Well, we don't want fibrin, which is like a mesh net, to be clogging up our blood vessels. So it's carried in the plasma in an inactive form called fibrinogen. When you learned about connective tissues in anatomy and physiology, you learned that there were 3 types of fibers. There are collagen fibers, reticular fibers, and elastic fibers. And then there was this little, and in blood, we have dissolved fibers. And these are the dissolved fibers. Fibrinogen is the soluble form. Fibrin is the insoluble form that comes out into solution. Okay. What other proteins do we have? Albumins. Albumin, it's a very important protein because this is going to be listed later on in another slide, but it holds water in your blood. Your blood is in your blood vessels, and albumin has a trap on that water, on this 91% of the plasma, and it helps to keep the water in the blood vessels because water can leave the blood vessels and go into the tissues. So it has an osmotic pull. It helps with oncotic pressure. It also acts as a pH buffer, and it carries certain things, certain hormones and bilirubin through the blood. The globulins, all of these, by the way, albumins, fibrinogen, and most of all the globulins are made by the liver. So if we have liver problems, we might have, or a kidney too, but we might have low levels of albumin, and then it's harder for us to keep our fluid volume in the actual blood vessels. If we have liver problems, we might not have all the globulins that we need, or we might not be able to clot because we're not producing fibrinogen along with some other things. So be aware that the liver makes most all of these proteins that are carried in your plasma. There is an exception on the globulins. We're going to have gamma globulins. That is our exception. The gamma globulins are our antibodies, and so they are for immunity. The other ones, we have globulins like alpha and beta globulins. They're going to carry substances through the blood. Like if I'm a hormone and I need a carrier, maybe I'll have an alpha carrier, alpha globulin carrier, or maybe I'll have a beta carrier. If I'm a vitamin, maybe I'll need to be transported through the blood attached to an alpha globulin. So these are all the parts to the blood, and it's sort of important. These are all the parts to the blood. 55% of a whole blood sample is plasma. Only 1 to 2% is made up of the buffy coat, which will be the leukocytes and the platelets. And then the hematocrit, which is made up of all the erythrocytes, will be about 45%. Here's 55%. There's a 45% down here of a whole blood sample. I'm talking too much. Sorry about that. Just happens. Okay, so then let's look at a hematocrit. Here we're drawing, taking a little bit of blood in a little capillary hematocrit tube. It's going to pull the blood up, and the centrifuge is going to circle it around and until all of the heaviest stuff goes to the bottom. So imagine this picture right here being in a very fine little tube, hematocrit tube. It will look like this. You will have a buffy coat. Here's a buffy coat. Here's a buffy coat. And look at the hematocrit. In females, notice how the hematocrit is less than the amount of red blood cells for males. About this much difference right here. Hematocrits are higher in males. Males have testosterone. That's one reason that they have more red blood cells. So higher hematocrit in males than females. In this upper part, the plasma, this is where we have those albumins. We have the different types of the globulins, everything that's dissolved, the ions, the hormones, all of the things. Okay, here are the functions of blood. Well, I'm pretty sure that you guys know that oxygen is carried in your blood. It's carried, you breathe in, oxygen goes to your lungs. It goes from the lungs, it diffuses into the blood, and then your oxygenated blood goes to the tissues, drops off. But here we go. Let's just do it this way. The heart, arteries lead away from the heart. There's a capillary bed. This is the site of gas exchange. And here's a vein going back to the heart. When the heart pumps this oxygenated blood that it received from the lungs, that oxygenated blood goes to the capillary beds. So we have high oxygen here, and this blood happens to be low carbon dioxide. And when we get to the capillary beds, these are in the tissues of the body, the oxygen will leave and go into the tissues. And the tissues make carbon dioxide when they're making ATP, and the carbon dioxide goes back into the blood and then back to the heart. So on this side, we're going to have low oxygen blood going back to the heart with a higher carbon dioxide load. So blood distributes oxygen to the tissues and carries carbon dioxide away. Also, any metabolic wastes that are made in the tissues will jump into this blood here in the capillary bed and be excreted either by the kidneys or it depends on what carbon dioxide would be one that we could actually expel through our lungs. We have hormones and nutrients that need to be distributed throughout the body. In the endocrine chapter, all of your endocrine organs secrete the hormones directly into the blood, so they need to be distributed. How? Through the blood. We haven't done the digestive system yet. The nutrients that we pick up from the digestive system are going to be going to the liver to be processed and then spread throughout the rest of the body. So distribution, that's one big role of blood. Another one is regulation. Regulates body temperature. We know when we're hot and our blood goes to our skin, it can dissipate heat, or if we're cold, our blood can be shunted inward to maintain our body temperature. So one big thing that our blood does because of the hypothalamic control is control our body temperature. Another thing, blood regulates our normal pH. Blood pH will be between 7.35 and 7.45. And it's regulated through quite a few things, but mainly the carbonic acid bicarbonate ion buffer system, water plus carbon dioxide. These things are in your blood. And if they combine together, then they form H2CO3, which is carbonic acid, which breaks down to release hydrogen ions and the bicarbonate ion. Okay, this is the bicarbonate ion, and this is the hydrogen ion. Well, how does our blood regulate its own pH through this bicarbonate carbonic acid buffer system? This goes in two directions. We're always in equilibrium. But if we add something on this side, it shifts these reactions to the right. If we add something over here, like hydrogen ions, it'll shift all these reactions to the left. And hopefully we don't have any, because of this bicarbonate ion, carbonic acid buffer system, which is in your blood. It's going to prevent radical shifts in pH by absorbing excess. pH is a measure of your hydrogen ions. And so if we add extra hydrogen ions, that's going to lower your blood pH, and we don't want to drop too low. And so how can we fix that? We have this buffer system. We can combine the hydrogen ion with the bicarbonate ion, produce carbonic acid, and then those hydrogen ions are bound up. And if we are producing lots of carbonic acid this way, then that carbonic acid will break down to form water and carbon dioxide, and we can breathe the carbon dioxide out. If, say, we're maintaining too much carbon dioxide because of our respiratory system, we're holding carbon dioxide in. Like I said, if we add something on this side of the equation, we shift over to the right, what's going to happen is we'll combine our water and our carbon dioxide to form carbonic acid, and then it will break down to release hydrogen ions into the blood and the bicarbonate ions, and this will also lower blood pH. So in that case, what do we do if this carbonic acid bicarbonate ion buffer system is overwhelmed? Then we're going to have to rely on our urinary system or our kidneys to try to get rid of those excess hydrogen ions. Okay. I think you guys know about the bicarbonate ion carbonic acid buffer system. We'll bring it up time and time again. Fluid volume. I said before, like especially with albumin, if we have enough albumin in our blood, it holds the fluid in the blood. And that's very important because here, when the blood gets to these capillary beds, they're just one cell layer thick. So if you're just one cell layer thick, it's easy for fluid to be forced out, but there is some osmotic pull by the albumin to hold the fluid inside of the blood vessel so we don't lose too much fluid to our tissues. Now, when we do lose the fluid to the tissues, by the way, it's going to be picked up by the lymphatic system and cleansed. So it's not like it stays there forever. But albumin helps us with fluid volume regulation. And last but not least, protection. Our blood protects us against blood loss just by having platelets that they help plug up the hole and start the whole process called coagulation. And then infection. Our blood protects us through our leukocytes, white blood cells from infection. So those are the three main functions of our blood. Now, what does the blood look like? If this is a sample of blood, everything in the back that you see here is plasma. It's just fluid. And you see these guys right here, these are the red blood cells. Everything that looks like that is a red blood cell. It's lighter in the middle because if we look from the side, it's a biconcave disc. This means concave going in. And it's also concave on that side. So we've got two little concave areas. So we describe red blood cells as a biconcave shaped disc. So because it's so thin in the middle and the light shining up through the microscope, it appears like this. All the red parts of your red blood cell are shoved out to the side, or the light can't penetrate it. And so that's why the red blood cells look a little whiter in the middle. These guys over here, there are a bunch of them clumped together, but these are all platelets. Here's a platelet. There's a fine example of a platelet, what platelets look like right there. And then these guys, now these are called red blood cells because they're red, right? And then these are our leukocytes, or white blood cells. Why are they called white blood cells if they have all this color to them? Well, they've been stained so we can actually see them. And we have 3 different types of white blood cells on here. Wish I would have marked over that one. But we have what's called a big old monocyte here. Notice how large it is compared to these two are the same guy, called a neutrophil. And here we have a little lymphocyte. And we'll get into these identifications in a subsequent lecture. But here we have blood cells, white blood cells, and platelets in our sample. Okay, we've pretty much gone over the slide already. The fact that plasma is 90% water, and then we have all these dissolved solutes in the liquid portion of blood called plasma, like the plasma proteins. Albumin acts as a transporter of the things, could be bilirubin, could be a hormone, acts as a pH buffer, because remember our blood pH is between 7.35 and 7.45, and this is one of the things that helps us absorb any massive changes in pH. And osmotic balance means it holds the water in the blood, so not too much of it goes out into the tissues. The other plasma proteins would be like the alpha and the beta globulins that transport nutrients, hormones through the blood. And the gamma globulins are actual antibodies, which we cover on later on in the lecture. Some non-protein, here the plasma, these solutes are classified as proteins. The non-proteins would be like the nitrogenous waste we talked about earlier. If you eat protein, you produce nitrogenous waste, and they'll be taken to the kidney, and we'll get rid of that nitrogen. Electrolytes is anything charged, like, where am I? Sodium or potassium, say. Respiratory gases would be oxygen, carbon dioxide. Hormones, anything that we covered in the endocrine chapter, would be floating around in the plasma portion of the blood, and nutrients from our digestive system. The formed elements are going to be the red blood cells, the white blood cells, and the platelets. Here's a normal hematocrit for males. You'll always have a scale. Here's a normal hematocrit for males, 47% plus or minus 5%, and females 42% plus or minus 5%, meaning males have a higher amount of red blood cells than females. And that is per cubic millimeter. I don't want you to waste any time learning these individual counts for red blood cells for females or males or infants or anything other than that. I assume that you can refer to a chart to find these normal red blood cell counts per. But notice that we're in the millions. And you saw that on a previous slide. When it comes to white blood cells, however, we're in the thousands per cubic millimeter. This is this. And we have about 150 to 500,000 platelets per cubic millimeter, or that's a microliter. Now, we're almost to the end of this little section. I am overheating, so I am trying my best to pause this recording for a second so I can breathe. Okay. All right. I get confused when I get hot. So here we go. Now we're going to talk about this idea of how our blood cells form. Let's see if I can't advance. That's okay. Some vocabulary for you. The term for the formation, poiesis, of blood is hematopoiesis. That's all of the formed elements. If someone says hematopoiesis, we're talking about red blood cells, formation of red blood cells, white blood cells, and also of the platelets. If you want to be specific, what is it called when we form just this red blood cell or just the platelets or all of these white blood cells? If we're forming just the red blood cells, it's called erythropoiesis. And if it's formation of white blood cells, it's called leukopoiesis. And if we're making the platelets, then we're talking about thrombopoiesis. And we'll compare and contrast on the next slide. But where is our blood formed? Where is it formed? You know this. It is formed in our bone marrow, in the red bone marrow in particular, right? And so in all of these cells, all of these cells down here on this little layer here, in this bracket, they're all formed inside of the bone marrow, the red bone marrow, and then they're put into the blood. But they all have a common origin. What is that common origin? It's called this thing right here, a hemocytoblast. Some people call it a hematopoietic stem cell because this is a stem cell. And this stem cells are cells that are undifferentiated. They're not specialized. And when they undergo reproduction, they can produce a wide variety of different types of cells. So this hemocytoblast lives in the red bone marrow, and most hematopoiesis is occurring in the proximal epiphyses of the humerus and the femur, and then mostly the flat bones like the sternum and the ribs. That's where this is going to occur. But we find these hemocytoblasts, and what they do is if they differentiate into a myeloid stem cell, then they can become either red blood cells, I'm all the way down at the bottom now, or platelets, or most of the white blood cells. If they differentiate into something called a lymphoid stem cell, then they give rise to just one type of white blood cell called the lymphocytes, although there are different versions of lymphocytes, like T lymphocytes and B lymphocytes, which convert into plasma cells, etc. And so, first of all, we have the hemocytoblast, and according to the chemicals it's exposed to, the signals it receives, is going to differentiate into a myeloid stem cell or into a lymphoid stem cell. And then, according to further stimuli, and we're going to see on the next slide the different types of hormones that it might be exposed to, this myeloid stem cell, if we just follow this myeloid stem cell, it can, if it goes down this pathway, it can form a red blood cell. Now, the biggest thing I want you to know about a red blood cell, look how the nucleus here, we have a nucleus and we have ribosomes, because later on in a subsequent lecture, we're going to learn that mostly inside of red blood cells, you just have hemoglobin. And hemoglobin is predominantly made of protein. And to make proteins, we need ribosomes, and we need rough ER and et cetera. So this myeloid stem cell that's going to differentiate into a red blood cell, as it goes through this path called erythropoiesis, it's going to be making all of the hemoglobin molecules that it needs. And because red blood cells are anucleate, meaning they do not have a nucleus, that would just take up space. They are anucleate. A means without a nucleus. Before they're ejected into your blood, before the red blood cells leave the bone marrow and actually migrate into the blood, they lose their nucleus. And so this is actually what's going to be put into the blood, something called a reticulocyte. This is an immature red blood cell. It should be about 1 to 2%. There should be about a 1 to 2% reticulocyte count. These young red blood cells, within a few days, will mature into this red blood cell, a mature red blood cell that you know and love. If you are like, oh, that's a red blood cell, that is a mature red blood cell down there. It only takes 1 to 2 days for a reticulocyte to become a red blood cell. Now, if this hemocytoblast differentiates into a myeloid stem cell and it goes down this pathway to become what's called a megakaryoblast, what it's going to do is it's going to differentiate into something called a megakaryocyte. Excuse me. Mega means large, kary is nucleus, and then cyte is cell. So it's a very large cell with a big, huge nucleus in it. This guy, this megakaryocyte, does not leave the bone marrow. It just rips little fragments off of itself, and these are the platelets. So the platelets are just like stamps being ripped off of. You guys probably don't even use stamps anymore. It doesn't really matter. The megakaryocyte never leaves the red bone marrow. Just little fragments of it are pulled off. So what you're seeing down here with these platelets are just cell fragments. They don't have a nucleus or anything like that, and they're going to have a short lifespan. Lifespan for red blood cells, we're talking like 4 months, 90 to 120 days. Platelets, we're talking maybe 9 to 11 days, depends on what's happening. But they've got a very short lifespan compared to red blood cells. If we go from the hemocytoblast to a myeloid stem cell, and then we go down this pathway, then we're going to produce all kinds of different types of white blood cells. And these are the white blood cells that are entering into the bloodstream. Everything on this row is entering into the bloodstream at a little immature form, and then within days or however long, hours, it becomes mature. And so this myeloblast, I'm not going to hold you guys accountable for all these terms, but this myeloblast differentiates into 3 different types of cells that are going to ultimately become our granulocytes, the basophil, the eosinophil, and the neutrophil, and then one type of agranulocyte. We go over these terms later, the monocyte. Notice how, oh, sorry, that's a different guy. This myeloid stem cell is slightly different pathway. When it goes down here to the monoblast, that's what differentiates into this agranulocyte, but that's okay. All of these guys from here over, all of these guys I just drew a line from, they're coming from the myeloid stem cell, which came from the hemocytoblast. If that hemocytoblast becomes a lymphoid stem cell, it differentiates into something called a lymphoblast. And those cells are what we know and love as lymphocytes in our blood. We'll get more specific later on. If you hear hematopoiesis, we're developing all of our blood cells. Erythropoiesis, just red blood cells. Leukopoiesis, just white blood cells, and thrombopoiesis, just platelets. And this guy right up here, this original stem cell, is under the influence of a variety of chemicals. What's going to determine whether it becomes a myeloid stem cell and then a proerythroblast, or a lymphoid stem cell and then a lymphoblast? That depends on what's happening in the body and what chemicals the stem cells receive. So here's hematopoiesis overall, formation of red blood cells. This is just a comparison. When we talk about erythropoiesis, there's going to be one hormone that's secreted, and we learned this in the endocrine system, erythropoietin. This is produced by the kidneys. Your kidneys make EPO, and that stimulates erythropoiesis. Now, why do the kidneys release erythropoietin? Well, if the kidneys perceive that there's a drop in oxygen levels, if there's a drop in oxygen levels, the kidneys produce erythropoietin, and we get the formation of more red blood cells. In adults, like I said before, hematopoiesis usually occurs in the red bone marrow of the axial skeleton, these flat bones, like, say, the sternum and the ribs, and the proximal heads of the femur and the humerus. In a fetus, when you were a little fetus, you could also make red blood cells in your liver, your spleen, and your yolk sac. Now, as an adult, we no longer have a yolk sac, but we still have a liver and a spleen. If, for some reason, our bone marrow can't make red blood cells, that our liver and spleen can revert back to that role of erythropoiesis. It's not a good sign. It's usually an indicator of underlying disease, and it's called, if your liver and spleen all of a sudden started making red blood cells because your bone marrow can't, it's called extramedullary hematopoiesis. It's an indicator of underlying disorder. So, all right, now to make our red blood cells, the kidneys release erythropoietin that prods along the production. And so if you have kidney problems, let me tell you, you might have production problems of your red blood cells. So the kidneys, we depend on the kidneys to produce erythropoietin, which stimulates erythropoiesis. And we also, if you have dietary problems, you might have a hard time producing red blood cells. Our main dietary requirements are the amino acids to produce the actual hemoglobin molecule. Lipids, carbs, B vitamins are always for mitosis. Vitamin A, copper, but keep in mind this iron. We'll come back to iron later on. And leukopoiesis, this middle column here, where we're producing our white blood cells, is stimulated by chemicals called interleukins, IL, and they're named interleukin 4, interleukin, etc., and colony stimulating factors. These guys, they're produced by such a wide variety of different types of cells. In erythropoiesis, the function of your red blood cells is to carry oxygen, so it makes sense if your oxygen levels drop that will trigger erythropoiesis. Your white blood cells are for immunity. So why would we want to trigger leukopoiesis? Well, if we're having, say, an infection, etc. So interleukins and colony stimulating factors, they'll stimulate different types of white blood cells to be produced, but they're produced by leukocytes themselves, by endothelial cells. These are the cells that are simple squamous cells lining the blood vessels, thymus cells, and bone marrow cells and astrocytes, even astrocytes. If you recall what astrocytes are, they are a type of neuroglia found in the central nervous system. And so even astrocytes can produce these interleukins and colony stimulating factors. So when do we stimulate leukopoiesis? When it's needed. You can have a very sharp increase in white blood cells if you have an infection. Now, these terms down here, leukocytosis and leukopenia. If you have a white blood cell count, a normal white blood cell count between 4,800 and 11,000 white blood cells per cubic millimeter. If you drop down this way, that means you have too few white blood cells, you would have trouble fighting infection. That is called leukopenia, because penia means weak, you don't have a lot of white blood cells. If you're fighting an infection and you increase your amount of leukopoiesis and you go above this 11,000 mark, that is called leukocytosis. This means a condition of white cells, too many white blood cells, but we need that when we're fighting off an infection. Thrombopoiesis, the production of platelets, is stimulated by a hormone called thrombopoietin. Just like thrombopoietin sounds like erythropoietin, this thrombopoietin stimulates thrombopoiesis, and it's made by the liver and in the kidney tubules. We make as many platelets as we need to. If we produce too few platelets, thrombocytopenia, I should write this down, we're going to have a hard time clotting. Some people have thrombocytopenia or a drop in their thrombocytes, platelets, after a viral infection. Sometimes that triggers that. If you have too many platelets, it causes the opposite. We don't want to have thrombocytosis. You don't want to have too many platelets because that indicates a problem and clotting can occur. So the platelets butt off the megakaryocytes, like I said, that are in the red bone marrow, and the platelets go right into the blood. And that's about it for this. We've already gone over the lifespan. Lifespan of erythrocytes, a few months up to 4 months is average lifespan. Leukocytes live hours to years, depending on what type of leukocyte it is. And platelets live approximately 11 days. For these, when I say that the white blood cells last hours to years, the hours, if you are a white blood cell and you're called into action, you might even last it's snowing again. You might only last just a little bit of time, not even hours, because you die for your cause, basically. All right, sorry for the long lecture. And we are done with the first part of the blood lectures.